வணக்கம். வெல்கம் டு NCERT Biology Reading. Chapter NCERT Biology Class 11, Chapter 14, 14.4 Aerobic Respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate is transported from the cytoplasm into the mitochondria. The crucial events in aerobic respiration are the complete oxidation of pyruvate by the stepwise removal of the hydrogen atoms leaving three molecules of carbon dioxide, the processing on the electrons removed as part of the hydrogen atoms to molecular oxygen, with simultaneous synthesis of ATP. What is interesting to note is that the first process takes place in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the pyruvate, which is formed by the glycolytic catabolism of carbohydrates in the cytosol. After it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by the pyruvic dehydrogenase. The reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes, including NAD plus and coenzyme A. Pyruvic acid plus coenzyme A plus NAD plus in presence of magnesium 2 plus in presence of the enzyme pyruvate dehydrogenase gives acetyl-CoA plus CO2 plus NADH plus H plus. During this process, two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid produced from one glucose molecule during glycolysis. The acetyl-CoA then enters a cyclic pathway, tricarboxylic acid cycle, more commonly called as Krebs cycle, after the scientist Hans Krebs was first elucidated it. 14.4.1 tricarboxylic acid cycle. The TCA cycle starts with the condensation of the acetyl group of the oxaloacetic acid OAA and water to yield citric acid. Figure 14.3. The reaction is catalyzed by the enzyme citrate synthase and a molecule of coenzyme A is released. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation leading to the formation of alpha ketoglutaric acid. And then succinyl-CoA. In the remaining steps of citric acid cycle, succinyl-CoA is oxidized to OAA, oxaloacetic acid, allowing the cycle to continue. During the conversion of succinyl-CoA to succinic acid to a molecule of GTP is synthesized. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with a simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycle where NAD plus is reduced to NADH plus H plus and 1 point where FAD plus is reduced to FADH2. The continued oxidation of acetyl-CoA via the TCA cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. In addition, it also requires regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows: pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ADP plus PI. In the area of mitochondrial matrix, which gives 3 carbon dioxide plus 4 NADH plus 4H plus FADH2 plus ATP. We have till now seen that glucose has been broken down to release carbon dioxide and 8 molecules of NADH plus H plus. 2 of FADH2 have been synthesized besides just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither oxygen has come into the picture nor the promised large number of ATP has yet been synthesized. Also, what is the role of the NADH plus H plus and FADH2 that is synthesized? Let us now understand the role of the oxygen in respiration and how ATP is synthesized. 14.4.2 Electron Transport System ETS and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on oxygen, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport system, ETS. Figure 14.4, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by an NADH dehydrogenase complex 1. NADH dehydrogenase is called complex 1, and electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 called complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electrons to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between complex 3 and 4. Complex 4 refers to the cytochrome C oxidase complex containing cytochromes A and A3 and two copper centers. When the electrons pass from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase complex 5. ATP synthase complex 5 for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process, yet the presence of oxygen is vital, since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor, unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation. In respiration too, in respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane-linked ATP synthesis as explained by chemiosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase complex 5. This complex consists of two major components, F1 and F0, figure 14.5. The F1 headpiece is a peripheral membrane protein. The F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which the proton cross in the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 4 hydrogen plus passes through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. 14.5 The respiratory balance sheet. It is possible to make calculations of the net gain of ATP for every glucose molecule oxidized. But in reality, this can remain only a theoretical exercise. These calculations can be made only on certain assumptions that there is a sequential, orderly pathway functioning, with one substrate forming the next with glycolysis, TCA cycle and ETS pathway following one after another. The NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. Only glucose is being respired. No other alternative substrates are entering in the pathway at any of the intermediary stages. But this kind of assumptions are not really valid in a living system. All pathways work simultaneously and do not take place one after another. Substrates enter the pathway and are withdrawn from it as and when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. Yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, there can be a net gain of 38 ATP molecules during aerobic respiration of 1 molecule of glucose. Now let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to carbon dioxide and H2O. In fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions. NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. 14.6 Amphibolic pathway. Glucose is the favored substance for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substances can also be respired. They would first be degraded to acetyl-CoA and enter the pathway. Glycerol would enter the pathway after being converted to PGAL, phosphoglyceraldehyde. The proteins would be degraded by proteases and the individual amino acids after deamination, depending on their structure, would enter the pathway at some stage with the Krebs cycle or even as pyruvate or acetyl-CoA. Since respiration involves breakdown of substrates, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. But is this understanding correct? We have discussed above, at which points in the respiratory pathway different substrates would enter if they were to be respired and used to derive energy. What is important to recognize is that these very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrates. Hence, fatty acids would be broken down to acetyl-CoA before entering the respiratory pathway when it is used as substrate. But when the organism needs to synthesize fatty acid, acetyl-CoA would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into a picture both during the breakdown and synthesis of fatty acids. Similarly, during breakdown and the synthesis of proteins too. Respiratory intermediates from the link breaking down process within the living organism is catabolism and synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one. 14.7 Respiratory quotient. Let us now look at another aspect of respiration. As you know, during aerobic respiration, oxygen is consumed and carbon dioxide is released. The ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed in respiration is called the respiratory quotient RQ or respiratory ratio. RQ is equal to volume of carbon dioxide evolved divided by volume of oxygen consumed. The respiratory quotient depends upon the type of respiratory substrate used during respiration. When carbohydrates are used as substrate and are completely oxidized, the respiratory quotient RQ will be 1 because the equal amounts of carbon dioxide and oxygen are evolved and consumed, respectively. As shown in the equation below, C6H12O6 plus 6O2 gives 6 carbon dioxide plus 6 H2O plus energy. Respiratory quotient RQ is equal to 6 carbon dioxide by 6 O2 gives 1. When fats are used in respiration, RQ is less than 1. Calculations for fatty acid tripalmitin, if used as substrate, is shown in 2 times C51 H98O6 plus 14 for 145 oxygen. The formula of tripalmitin is C51H98O6 tripalmitin, which gives 102 carbon dioxide plus 98 H2O plus energy. Respiratory quotient is 102 carbon dioxide by 145 oxygen equal to 0.7. When proteins are respiratory substrates, the ratio would be about 0.9. What is important to recognize is that in living organisms, respiratory substrates are often more than one. Pure proteins or fats are never used as respiratory substrates. Then, summary: plants, unlike animals, have no special system for breathing or gaseous exchange. Stomata and lenticels allow gaseous exchange by diffusion. Almost all living cells in a plant have their surface exposed to air. The breaking of carbon-carbon bonds of complex organic molecules by oxidation cells leading to the release of a lot of energy is called cellular respiration. Glucose is the favored substrate for respiration. Fats and proteins can also be broken down to yield energy. The initial stage of cellular respiration takes place in the cytoplasm. Each glucose molecule is broken through a series of enzyme catalyzed reaction into two molecules of pyruvic acid. This process is called glycolysis. The fate of the pyruvate depends on the availability of oxygen in the organism. Under anaerobic condition, either lactic acid fermentation or alcohol fermentation occurs. Fermentation takes place under anaerobic conditions in many prokaryotes, unicellular eukaryotes, and in germinating seeds. In eukaryotic organisms, aerobic respiration occurs in the presence of oxygen. Pyruvic acid is transported into the mitochondria where it is converted into acetyl CoA. With the release of carbon dioxide, acetyl CoA then enters the tricarboxylic acid pathway or Krebs cycle, operating in the matrix of the mitochondria. NADH plus H plus and FADH2 are generated in the Krebs cycle. The energy in these molecules will, as well as that in the NADH plus H plus synthesized during glycolysis, are used to synthesize ATP. This accomplished through a system of electron carriers called the electron transport system ETS, located on the inner membrane of the mitochondria. The electrons, as they move through the system, release enough energy that are trapped to synthesize ATP. This is called oxidative phosphorylation. In this process, oxygen is the ultimate acceptor of electrons and it gets reduced to water. The respiratory pathway is an amphibolic pathway as it involves both anabolism and catabolism. The respiratory quotient depends upon the type of respiratory substrate used during respiration. Thank you.